Receptor tyrosine kinases, RTKs. RTKs are a class of enzyme-coupled receptors. Activation of RTKs and the signal transduction pathways they activate lead to the following responses: cell growth and proliferation, cell division, survival, anti-apoptotic, angiogenesis, formation of blood vessels, changes in cell shape and migration through cytoskeleton rearrangement, differentiation. Note, RTKs and/or their respective pathways are hyperactive in malignant, aka cancer cells. Here's a table showing some of the signaling molecules in receptor tyrosine kinase pathways, the receptor families, and the type of responses that will be garnered from activating these type of receptors. As you can see by the responses, a lot of the responses are cell survival, growth, proliferation, and RTK has a ligand binding domain that is extracellular, and a tyrosine kinase domain that is on the intracellular part. What does a tyrosine kinase do? Tyrosine kinase will catalyze the reaction of phosphorylating a tyrosine using ATP. So there's also serine and threonine kinases, and of course the tyrosine kinase that we're talking about now, and the phosphate is added to the free OH groups of the R groups of these amino acids. There are two methods by which RTKs are activated. RTK monomers form dimers in the cell membrane, and the binding of signaling molecule leads to the activation of the intracellular kinase domain. So you have the monomers that are not together, they haven't formed a dimer, and you have the signaling molecules. You have formation of a dimer, and then binding of the signaling molecule, and that leads to activation. And once the receptor is activated, you have this trans-autophosphorylation event that occurs where each tail phosphorylates the other tail. An example of this type of activation is the epidermal growth factor receptor, EGFR. Activation of EGFR promotes cell cycle progression. In many cancers, EGFR is overexpressed. Thus, there is an increased concentration of EGFR on the cell membrane. This overexpression leads to an increase in growth and division. Cell cycle progression signals being received by the cell, even though the amount of epidermal growth factor is not increased in the extracellular compartment. How? Let R be a monomer of EGFR, the receptor, and let L be the signaling molecule, epidermal growth factor. The Ka for dimerization would be the dimer over R times R, the concentration of R times the concentration of R. Now the Ka for the dimer binding the ligand would be the dimer-ligand complex concentration over the concentration of free dimer and concentration of unbound ligand. Now, if the concentration of R increases because of some sort of mutation that leads to increased expression of the receptor by a factor of Y, where Y is larger than 1, then the dimer concentration will increase by Y squared. Well, if the concentration of the dimer increased, then the concentration of dimer ligand will also increase by the same amount. If the dimer ligand complex is increased by a factor of Y squared, then the growth and division signals a cell is receiving will also increase proportionally. So the main thing here is that by increasing the concentration of the receptor, even though the concentration of the ligand, the growth factor, is not increasing, you will still get an increase in growth signals being received by the cell. Method 2. An RTK monomer binding of its signaling molecule leads to the dimerization and activation of the kinase domain. So in this case, you have the two monomers which are not together, and you have the signaling molecule, same as before. You have the signaling molecule binding to a single monomer, and that will sort of push for the monomer to form a dimer. So the binding of the signaling molecule to the monomer increases the probability of dimer formation, or improves the thermodynamics of dimer formation. As when formed, you get activation of the receptor, the tyrosine kinase domains are activated, and you have trans-autophosphorylation where the two tails will go ahead and phosphorylate each other. An example of this type of activation is the human MET receptor, whose activation promotes cell cycle progression. It can be the case that dimerization is sufficient for activation of kinase domains. The MET receptor is overexpressed in many cancers. Increased membrane concentration of the receptor leads to dimerization and activation in absence of signaling molecule, hepatitis growth factor HGF. In this case, If a dimer forms, even though the signaling molecule is not bound, you will get activation. The role of the signaling molecule in a case like this is that the signaling molecule will promote the formation of a dimer. But if you have increased expression because of a mutation of the receptor, then you're going to run into the same problem we ran into before because you have a Ka for dimer formation. And if you increase the amount of R by some factor Y, you will have to increase the amount of dimer by the same amount, so you're going to have more dimer formation. But if you have more dimers forming, that means you have more activated receptors, and that will lead to this receptor being activated and sending the growth signal into the cell, activating the signal transduction pathway that promotes growth. If the dimer concentration is increased by a factor of Y squared, then the growth and division signals the cell is receiving will also increase proportionately. The phosphorylated tyrosine of the receptor's intracellular tail act as binding sites for intracellular proteins in the pathway. Once the receptor is activated and the tails become phosphorylated through trans-autophosphorylation, phosphorylated tails create docking sites for other proteins to bind to, proteins that are part of the signal transduction pathway. Intracellular signaling proteins which bind to oligopeptides containing phosphotyrosines have specific binding domains such as SH2 domain or PTB domain. Both protein domains will bind to phosphotyrosines.